Okay, thank you, Nora, for this introduction. It is a great pleasure to be here to learn from the families. And we teamed up, I would say, because we do believe that AGO1 and AGO2 genes actually cause a very similar, if not exactly identical syndrome. And what we want to show you today is first, want to share our knowledge with you. What do we know about these syndromes? What does it mean for families, for affected individuals? But then also to show you where the path is going, what we are planning to do to better understand this disease, both in terms of the clinical course, then also better understanding of the molecular mechanism of this disease, and hopefully bring the path towards a therapy that would at least ameliorate some of the symptoms. Yeah. Okay, so just to give you a little background on the genetics. So the DNA is actually present in all cells of our body. If you stretch the DNA, it is as long as a football field. And it consists of several chromosomes, 46 chromosomes. On each of these chromosomes, there are many, many genes. And today we know that there are around 20 genes, and all of this genetic information is filled up from 4 letters, A, T, C, and G. So this is this whole stretch, as long as a football field, is actually built of these 4 letters. Seems very simple, but actually it makes a rather difficult and complicated genetic information that is mostly inherited from our ancestors. Many things that we have, the way how we behave, these are all inherited. And then among some of the inherited things are also some variants that make them who we are. And then there are also some variants or mutations which arise de novo, which are not present in our ancestors, which are not present in the mother and the father. And these are the ones that we'll be mostly talking about. And what we also know is that more than 7,000 genes out of the 20,000 have since today been connected to a monogenic disease. And when I say monogenic disease, is that this one mutation really leads to a disease. It's that one single alteration. It's in most of the cases, one of these letters is changed, and it causes a disease. So if you look at it from such a perspective, it's quite crazy. But that's just the way it is. So what are the neurodevelopmental disorders? These are very rare disorders, and there are several symptoms that we observe, like intellectual disability, motor developmental disability, speech and language disability, autism spectrum disorders, attention deficit hyperactivity disorders, many various types of epilepsy. And what's interesting is that in many cases, we see a combination of these symptoms. And what we know is that the major cause of these neurodevelopmental diseases are so-called de novo mutations. What these are? These are alterations that are present for the first time in the individual. They are not inherited by the father, they're not inherited by the mother. And I think it is very important to state here that if your child has such de novo mutation, there is absolutely nothing that any of you could have done wrong. So according to the knowledge that we have today, if you drank 2 or 3 glasses of wine, it did not cause a de novo mutation. If you smoke, it did not cause a de novo mutation. We do not understand. What we know is that this de novo mutation happened in each of us. It's just a question, where do they happen? We know from the large sequencing studies that each of us has 70 novel variants across the whole of the genome. It's a kind of an evolution going on. But if you have a de novo mutation in a critical gene, in a critical region, that this mutation can cause a syndrome. And it can be either as a result of mutation in a germ cell, either in egg or sperm. And this is something to what we refer as gonadal mosaicism. And it is a really mean thing because we cannot test it. So if we identify the de novo mutation in your child, we cannot go on and see if you have a gonadal mosaicism. But it means that there is a slight chance that your second child would also have the identical de novo mutation as the first child. But in the majority of the cases, it's not inherited, it's not in the germ cells, and it actually occurred during the first meiosis. What is the first meiosis? It's the time point where the egg and sperm connect and only one cell forms. And the first division of this cell into the two new cells is called meiosis.
Yeah, okay. So before I will continue, I just wanted to say one word because I'm so glad to be here with you. It was really a nice moment this morning, really emotional for me. I think emotional for you also. So I didn't know some of the family with AGO1 mutation, but I knew a bit them through the publications, through the description of the children. And so I'm really, really glad to be here and to meet you personally, even if I could not meet as a children today, but it's really, really a big pleasure and really, yeah, emotional for us. I'm so admirative of what you are doing all. I know that you have no choice, you have no choice, but still you're amazing. So yeah, so I'm really glad to be with you. So I will try to, to follow up a bit then. So it was just to represent you a bit about brain development. So you know that maybe the cell unit of the brain that is really important for brain functioning is the neuron. So you can see here a neuron that will connect to another neuron. And the idea is one neuron will pass some information through this kind of junction between the two neurons that we call the synapse. I'm sorry, in French. And the synapse is like a river that you will cross to pass the information between neurons. And this is like how brain is functioning. And so our goal is to try to understand how the mutation identified in the different children could affect at some point brain development and brain functioning with the transmission and information between the different neurons. So it's just to show you really like a basic view of what is important for brain development. So we have several steps important for brain development. I won't go into the detail, but basically we have neural stem cells that will proliferate and that will then differentiate and make the neuron. So this cell unit of the brain, and the neuron will connect together to form what we have like the normal brain, the adult brain we have here. And if something goes wrong, it could be environmental, but it could be also genetic. In the case, the talk we are talking in the case of your children and in the case of what we are talking today, it's a genetic thing that happens. So an impairment of the function here that will affect some ways the brain development and the brain functioning. And it's really not so easy to understand what the mutations are doing on the brain function, and we will try to present you a bit about that today. So just before going to the consequences of mutation, just as Davor told you, that we have the human genome here, so you can see all the different chromosomes. So you have 20 pairs of chromosomes plus the sexual chromosome, and it's like a big book. So I have several big books where you can find some information that will be useful to form the protein and to make the cells function, and the neuron also, but also all the different types of cells in the body. And so here is a representation of what you can find in one of the pages of this book. So you have all these different, all these sentences. So there is 4 different letters that are used, A, T, G, C, and that are like following each other. And some of this part of this DNA are important to make a protein and to make a protein functional. And so I don't know, but maybe a lot of you heard about the messenger RNA with the last in the last 3 years. And so what is a messenger RNA? Just a photocopy of this information, a book of information contained by the book that will allow to build a protein. So you can imagine that a protein has a specific function in the cell. It could be like, for instance, in a shelf where you put some books. And so you really need to have some information here, the correct information to build a correct protein and to have a shell that can contain a book. And so we put this slide maybe to explain you, because I don't know how many of you had like the genetic record where you can find all this information, the seed point or peak point. I don't know how many of you have this record. Maybe you have it and you don't really remember of it, I don't know. So I just to explain you, so you have here the genetic information there, and if something is changing, so if there is a mutation in the genetic information, it could have different consequences. So I won't talk about the deletion, but imagine you can have like I will more talk about this category, because all the mutation in AGO1 and AGO2 fall in this category. So there is here a change in one specific letter of the DNA, and this will be sufficient to change one part, but only one small part of the protein, AGO1 or AGO2. And this could be sufficient to alter the protein function meaningfully. And so what you could find in your medical record is like information, genetic information, saying that at the position 11 of the genes, you will have the letter that was a C that will become a G. And this is the consequences at the protein level, at the AGO1 or AGO2 protein level. So you have an amino acid that is called a serine, for instance, that will be changed in a cysteine at the 4th position of the protein. So in case you find your genetic record, you could see which kind of mutation you have. And why I'm saying that is because for AGO1 and AGO2 related neurodevelopmental disorder, there are some recurrent mutations. So some of you can share, for instance, maybe your children share the same mutations that another children. And so if you want to see that, you can just have a look at this information there. So the amino acid, the number, and the change. And so just to tell you that so all the proteins that are really important here for the different steps of brain development or brain functioning, so the transmission of information between neurons, they could be mutated. So they could exist a mutation in the DNA, and that could impair maybe the function, and that would lead to a neurodevelopmental disorder. And that's why we know that now we have more than 1,500 genes that could lead to a neurodevelopmental disorder when they are mutated. So each of them represents a rare disease, but if you put everything together, there are a lot of rare diseases, so it's not so rare finally.
Okay, as Amelie said, what we first identified were 20 missense mutations in patients in AGO2 gene. What is interesting is that some of them were recurrent. It means that they were not identified only in one patient, but in 2 patients. And this is the reason why, in order for you to be able to see which mutation does your child have, is the reason why Amelie was explaining you how you can find this information. So we identified a deletion of one phenylalanine in a child from Singapore, and I identified in FIN this amino acid substitution missense variant, and then later on we identified exactly the same missense variant in a girl from Seattle. Then we have at the position 201, we have a boy from Leipzig and 2 children from the Netherlands. And there is exactly the same amino acid which is affected, but the substitution is different. Then we have several children who have this variant. This is also a recurrent variant. This variant was identified in the girl in Silvana, who was also here. And then we also identified here a mutation in monozygotic twins in children from New York, and also one from North Carolina in the United States. And the question is, what are the typical, what is the clinical spectrum of this disorder? The first time when I identified the first TARGET2 mutation, FIN, I was rather lucky because I connected with colleagues in Netherlands, whom I knew that they are sequencing a lot of similar patients. So we identified 3 more patients, and therefore I could tell Antje a little bit about this disease, that there are 4 children, including Finn, but I wasn't able to actually tell her more than just the gene name. You know, I couldn't tell her what the prognosis, what is the life expectancy, if there are any special therapies that would help her child. This was all unknown. And it takes a lot of time to really get this good clinical information. Because what we do is I get contacted by a colleague, I send this colleague a clinical table. I ask this colleague also to send me a clinical description of the course of the disease. But in the most cases, the only thing that I receive is a clinical table. So what I get is something like this. I get the information if the children have intellectual disability, and all of them have it. The only question is to which degree. And we really observe high clinical variability in these symptoms. There are children who have a mild intellectual disability or severe intellectual disability. But also this information is really hard to really interpret because this is a subjective thing. We also know that most of the children have motor developmental delay. They have impaired speech development. They have impaired receptive language. But also there is a huge variability. Some children speak first words at the age of 20 months. Some children speak only 2 words at the age of 5 years. We also know that many children have muscle weakness, muscular hypotonia. We know that many of them develop several autistic features, including self-aggression and aggression to the others. But we don't know really if this is something that happens at a certain time point. Is it something that happens only after puberty? Or does it occur in some children already at the age of 3? This is the information that I do not receive from your medical doctors who follow up your children. We know that many children develop seizures. This is also problematic because I never receive an information which kind of medication was given to our children. Did they respond? Or they didn't respond to some medications. And I'm telling you this because we need your help so that we can better understand this disease. So in order to do this, Dr. Ilicheva is here today and he'll be joining our team and she will help me understand this. And I really ask you, both present here and also those online, to help us understand the disease of your children. That's the only way, by doing the interviews with you, is the only way how we can really understand this disease. Because we do not receive the critical data from your medical doctors. And also there are some things that we can only learn from you. There are some things that they don't even know. For example, I thought for a long time that there was actually only 1 patient who has a premature puberty. And today I learned that there are at least 2 more. Because the colleague did not tell me that. Moving on, we know that many of the children had feeding difficulties during neonatal period. But also this is hard to interpret. What does it really mean? And you are the ones who want to ask me this. But this is the only information that I received from the colleagues, that they have neonatal feeding difficulties. Were they muscles so weak so that they couldn't suck? Were they so agitated that they didn't want to suck? I don't know this information. And I need your help. Again, one of the problems that we are experiencing when we are trying to better understand this disease is that you observe that there is a huge variable expressivity. It is from mild to moderate to severe. And even children who actually have the identical mutation, we observe even in those children the variability. And this is something that bothers us both a little bit, much. And we still cannot completely understand this. So what we have also observed is that there seem to be some symptoms which are specific to corresponding mutation. For example, both patients who have lysine to proline at the position 192 had breathing problems in the neonatal phase. They had central apnea. All three patients who had this missense variant had the bilateral clinodactyly of the fifth finger. This is a curvature of this fifth finger. They had misaligned teeth with large incisors. And this information is extremely helpful for us because it gives us actually a molecular hint that these missense variants could actually be affecting the function of the protein in a slightly different manner. That's why it is extremely important for us to get accurate and as good as possible clinical data. And then again, there are some symptoms which are observed only in single individuals, and we can't really be sure if they are due to this specific mutation. For example, in Silvana, we observed blue sclera, so the part of the eye that should be actually, it is always white, it's bluish. This patient had a problem with kidneys. And this is what I already said. I thought this was the only patient who has a premature puberty. Then I learned that I was wrong. So how can we explain these clinical differences? And this was one question that we already received from Nora. One explanation is that the missense variants differently impact the function of the protein. Then there could be that there are some other genetic alterations which have not been identified, which are perhaps even unknown, which do have an effect on the whole clinical picture. There could be a combination of both. There could even perhaps be some environmental factors. We're not sure.
So just to say one more about the mutation in AGO1, so you can see here all the different mutations that were identified. I tried to put here, but it's a really non-exhaustive list and it's difficult to see the number of families we are aware of in the different countries. So you can see that in the United States, you have 14, for instance, already family, 2 in Canada, 9 in France. They are not there today, but yeah, 9 here in France and other in Netherlands, Germany, Switzerland, Romania, Italy, Spain, like kind of every European country. I think this number will really grow a lot in the next months or year. Just to show you, because so Davor was talking about maybe the different mutations have different effects on the protein, so I won't go into the detail, but you can see here in the different colors the different functional domain of the protein. And all the mutations are not located in the same domain. So maybe mutation located here in the protein could have a different effect from the mutation located here. And it's still something we don't know, and we are still working on to understand a bit if it's the case or not. I wanted to show also so the clinical picture of the patient, and so it's just to prove you that it's really it has a lot in common with the AGO2 syndrome also. So you can see that the most frequently clinical manifestation is developmental delay and intellectual disability present in all the children, and it, as for AGO2 syndrome, it's also high variability in the degree. It could be mild intellectual disability or severe intellectual disability. The speech impairment is also really present in all the children, and you have after some other clinical signs that are kind of recurrent in children, behavior problem such as anxiety and also aggressiveness that Barbara told about. You can see also motor delay, autism spectrum disorder or autism behavior here. You have some sleeping disturbance that were reported, so I don't know if it's the case also for AGO2, but yes. And it could really impair the quality of life of the family. So it's something we also need to know and to take into consideration. And you have also other clinical features. For instance, you can find some seizures, but only in half of the children, and we still don't know why. The muscular weakness also that were able for AGO2. So finally, when you see that, you cannot see really, if you see the table, you cannot really see AGO1 or AGO2 related neurodevelopmental disorder. So we really think that because the mutation are the same and the clinical manifestation are the same, that this syndrome are really closely related. So you can see here the picture of some of the children. So I think you can recognize some of your children here. So there is Ashton, for instance. And there is also, so I would say hi to Oli and Naomi here because I know that she is present online. And so yeah, you can see that they share some particular trait of the face between the different children, and this is also shared by the children with mutation in AGO2.
Okay, so once Amelie's manuscript about the AGO1 mutations came out, what we observed is that there are several of the mutations which are almost exactly the same. So you have to bear in mind that the AGO2 gene is 2 amino acids shorter than AGO2. So, for example, the deletion at the position 182 actually corresponds to 180. The missense variant at the position 192 corresponds to 190. And so there are several variants which are really identical between AGO2 and AGO1. That's why when you also compare the clinical features, the common clinical features that we have access to, we really believe that this might be very similar, if not identical syndrome. And again, we can learn a lot from the clinical course of disease, and it can give us a hint towards the molecular mechanism. Meaning there are several functions that AGO1 and AGO2 share. There are also some functions which are exclusive for AGO1 or exclusive for AGO2. So if our hypothesis is correct, this is really identical syndrome, then the molecular mechanism causing the AGO2 and AGO1 should actually be the same. That's why it's very important for us to get as good clinical information as possible. So, this being said, the question is, what is our ongoing research? What are the future perspectives? So first, what we want to do is we want to really understand the clinical course. We want to understand this genotype-phenotype correlation, which means, is there a difference how the mutations affect the protein function on one hand, and is it correlated with perhaps different clinical groups? And therefore, we're also trying to establish a patient registry. The next scenario, what we want to do, we want to analyze the cells of the patients which are primarily affected. These are the neurons patients. And obviously, we just can't take a little piece of the brain of the children. So we have to find another way how to generate the neurons. I'll show you how we do it. Then we are working on several animal models. Amelie's collaborating with a colleague who is working on worms. Professor Kranenkamp has established mouse models. Little bit of We want to analyze the impact of these mutations that were identified on diverse functions of both AGO1 and AGO2. And also we hope to establish a sensitive functional assay, which will on one hand enable us to prove the diagnosis, on the other hand, enable us to do some screenings for potential therapeutic targets. So,
I think it's kind of maybe relevant with what you say, that we are now trying to better characterize the clinical consequences of AGO mutation. So I put AGO there. So I really encourage all the family members with AGO1 mutation to participate to the clinical registration that they will do with DAVR, because it would be really important to see the common things and maybe the particular thing if it's there are some particularity related to AGO1 or AGO2. So yeah, we need to study the consequences of mutation to understand what they are doing on AGO function, and also what to understand how altering this AGO protein, AGO1 and AGO2, could impact brain development and brain function. And I have to say that with the work I've already done to try to understand that, for now I still don't have the answer of what the mutation are clearly doing on AGO function. And so the direct benefit could be like to better diagnose the other children affected, for instance, by AGO1 and AGO2 mutations. So if we consider both syndromes together, there are more than 70 families that have been identified so far. And so I think it's a kind of frequent rare disease. So we should identify more families in the next coming month, and it will be improved by better know the clinical manifestation associated with the syndrome. So also identifying novel mutations could also help us to better understand the disease mechanism, because if we have more mutation on the protein, we could see what are the positions that are really important for the protein function. And in the future, for sure, it will also maybe help to identify potential therapeutic targets. So that's the idea, basically.
Okay, so how do we want to delineate this clinical course and genotype-phenotype relation? And I should have put AGO1 and AGO2 here. So what we want to do is interviews with family members. This is, as I said, something that Dr. Lesicev and I will be doing together. We want to revise the clinical imaging and blood values. So we would be happy if we would receive as much clinical and medical records that you are willing to share with us. And we would then make a patient registry in the hope that we would really understand these syndromes in a better way. We are always looking for new patients, and we have established a Facebook group, and for a long time, Antje and I were the only members of this Facebook group. Then luckily Nora came and took it over and brought it to a next level. But also with the help of Facebook group, we identified some new patients, because not every colleague who identifies an AGO2 variant will contact us, especially if the variant has already been published, because then it's clear for the colleague that this is the cause, and there is actually not much of a reason for contacting us. In most of the cases, we are contacted if a colleague identifies a novel variant, which has not been identified before, because in such a case, the colleagues are not sure if they could be causative or not. And this is also one of the reasons why we hope that also through Facebook group, we would learn more about patients who have especially variants that have already been published.
Can I ask a question? Because our son, he doesn't have a mutation in the AGO gene, but he's missing
the first 3 exons. Yeah, I know.
You know? Okay. It's a bit different than
I haven't put it because this is a very special case.
Oh, okay.
So I didn't want to. No. It is in terms of genetics, how this impacts the protein function. It's a bit of a special case, and I did not want to disturb the audience with this.
Okay.
But just for your knowledge and for the knowledge of others, the operation mutation was identified is actually has the similar effect to this variant here. So you belong to the group. Okay, so how do we want to further analyze the molecular mechanisms? Something that I'm going to say again.
We said just a few words about the models we can use to try to study the effect of evolution. So one, there are the cellular model on one side and the animal model on the other side. And so we will have some example of both, of course, what we can do with cellular model and what we can do with the animal model. So with cellular model, what you can do, you can make the protein with the mutation expressed in one cell to try to follow, to understand what this protein with the mutation is doing to the cell. And so we can perform all this kind of study. It could be in a regular cell, or it could be also in some special cells that we talk about. And so maybe you already heard about it, but we can take some fibroblasts of patients, for instance, and reprogram them in order that they could make some neurons. So it's like in vitro. So we will put the cell in vitro. We will act different reagent and factors, and we can obtain neural stem cells that could be differentiated into neurons, and also that could be differentiated at what we call cerebral organoid. It's like mini brain in vitro. It's not a brain at all, but it's like there are some neurons, some connection. And this could be interesting because we can here study different things compared to cells that were obtained without the mutation to see what are impaired at a different step of neuronal differentiation that could represent brain development. And so in this kind of different cellular model, we can do some molecular and cellular study to understand the mutation consequences. And we can also use this model to test therapeutic approaches when we will have identified some potential pharmacological or genetical therapeutic approach. So maybe you want, or I can say one word of the models that were already developed by Harlem in Strasbourg or in Ago. So for AGO2, there were some primary fibroblasts that were they differentiate to create this kind of cell that could be differentiated into neurons. And there are 3 cell lines already established. So you can see here the different mutation and the fibroblasts they are coming from. And for AGO1, we didn't differentiate from fibroblasts, but we introduced in the cell this mutation. So we've seen 199 serines, that will be some of you have. So I took this one because it's one of the most recurrent one, and also because of Oli a bit. And so we currently we will work on this cell. So we are generating this cell, and we could do the different analyses and differentiate them into neurons and go and do this kind of analysis. I don't know if you want to introduce that because you will need it in a way,
or I could just write it. Maybe.
Okay.
Sorry, we haven't trained this part. So what we want actually to do is we want to understand what's happening in the neurons of your children. That's why we are doing this. As I said before, we cannot just take some parts of the brain of our children. So what we want to do, we want to understand what changes are happening in specifically in their neurons and on this RNA level. And also there are some special kinds of RNA, which are called microRNA. And we know that both of these genes, in a very complicated manner, actually regulate also this micro, like very small RNAs. And this is what we want to see. We want to see when does it happen? Are the changes happening already at the very beginning of the embryogenesis? Are they happening a little bit later? Or are they happening when the neurons are already formed? We are also doing several characterization of these neurons. We want to see where they are they long enough? Are there any changes there? Can they work together? Because these neurons, they have to be in contact to each other through this synapse. So is this synapse working? These are all the analysis that we can only do in the neurons. We can't do them from blood, we can't do them from skin biopsy. That's why we are using this technique that has been developed in the last years, which we are implementing in both of our labs to better understand this disease. What you're also trying to understand is what this proteins, like almost all proteins in our body, don't have only one function, but they have several functions. And we are trying to analyze some of these functions in our labs. We're trying to analyze some of these functions in close collaboration with Gunther. And we are also looking for other colleagues in the field. That's why this was a perfect opportunity here to meet with other scientists from the organoid field and talk to them and see who of them can help them understand these diseases better. So we are also collaborating with a group from UK. We hope to perhaps also work with a colleague here from Roche. Antje has already found a colleague that from Airbus Special Program that might help us build this brain organoids. So we are actively trying to find help from scientists all over the world. And now I'll turn this to my colleague. Thank you.
So finally, I want to also say some words about what we're doing to functionally characterize what's wrong in the patients. And one aspect that we do is that we take a virus system to bring AGO2 protein into neurons. And by doing so, we can choose to take the wild type, so the normal, the non-mutated, or we can mutate it. And Deborah is here. She did this for some variants or some mutants, which are listed here. And we have made a virus to express this and use this virus to express, so to put the protein into neurons. And we want to see what's going wrong in the neurons. And one aspect of this will be that we isolate AGO2 from these neurons and see what kind of RNAs are attached to it. And this is a biochemical approach. And I make a living by teaching biochemistry to medical students. And that's sometimes difficult. And if I can do it to medical students, I can do it to you guys. No problem. So we go through this for a second. Neurons are cells. And all the cells in my body and in your body are little factories producing protein. They use the information on the genes, on the genome, on the chromosomes to make those proteins. And the intermediate is the mRNA. So we have, anyway, we have the gene, we have the mRNA, and we have the protein. The protein could be AGO, but there's many other genes, there's many other proteins. If you were a factory, not for proteins, but if you were a factory for cars, you would make wheels and you would make an engine. And if you plan it properly, you would make one engine and 4 wheels for every car you want to produce. Our cells have to organize it the same way. So they go from the gene for motors, for engines, and they do not make so much mRNA for it. They just make, moderate. But the gene for wheels, they have to make more mRNA for it because they need more wheels than engines. But this is not so precise. But our cells need to be precise. So they just get the information, maybe, yeah, we need more mRNA for wheels. That's okay. But then to fine tune this, to make it accurate, we need microRNAs. These are not listed here, but I think you've heard the term maybe during discussions. So microRNAs will then affect mRNAs, and microRNAs will just reduce. Say, we really don't need that much of the engine. We just need 4 wheels, not more. And they will just adjust it properly. So the microRNAs are the fine tuners of gene expression in a way. And microRNAs rely on the AGOs. So the AGO plus the microRNA tells this mRNA not so much, and tells another mRNA we're too much. So we have to organize this properly. So if you don't have an Argonaut, it will get completely out of balance. You just produce engines, but no wheels or whatever, no cars. So that'll be a disaster. I guess our assumption is that if your Argonaut is mutated, it will work in a way, but it will not work so precisely. And then you get kind of a mess up. The balance of proteins is not really correct, kind of correct, but so
just 3 wheels.
3 weeks. Like, too many. We should do. You know, you're right. Maybe somebody has tried it. But it would do, but it's not so perfect. And it would give you a difficulty in running around, you know, competing with other motorists. So that's a problem. And this is what we think. This is one hypothesis. This is one assumption that could go wrong with the Argonauts. And for this, we would really like to isolate Argonaut, the normal one and the mutant, from neurons and see what kind of microRNAs are attached to it. Because there's many microRNAs, there's also many mRNAs, and this regulation is a very, very complex thing. So one idea is to isolate this, identify the microRNAs and see what's changed, then get an idea what's going on. So Deborah does all this. She expresses in neurons, and you see the GFP. This is green. This is where we have something expressed. And if we do this with the ARGO2, with the wild type, it really does that. It goes into, this is just a very tiny extension of the neuron. And we have these little bodies in there called P bodies. This is where Argo is supposed to go, and it does go there. So we have a good model system to isolate our GFP Argo2 from this and get the right information on this. This is one thing. This is one approach that we're taking. The other approach is animal models. There are people here in the room sitting in the back. Welcome. We do this in nematodes, in worms, because the whole idea that there is something like Argonaut and that there is something like microRNA comes from these model organisms, nematodes, also from Drosophila, also from plants. And only later on we learn that this also happens in humans and in mice. So nematodes are very precious model organisms in this respect. And when in animal models, if you bring in the changed Argonaut, then you may see a phenotype, let's say motor dysfunction, there is something wrong with the head or whatever. And maybe you can also do behavior, learning, memory studies, social interaction. I don't know, nematodes, do they interact? Do they have social life? See, you need to study it. And then we can have get interesting conclusions from these things. But also we can go into mice. My system of choice here is the mouse. And we have put 3 variants which are relevant into the mice. And really it's a collaboration with Davor. Davor does all the sequencing, if it's really in there or not, and confirms this. So we have the leucine 192 protein mutation in mice. We have this 733 mutation in the mouse. And we also, by chance, generated a mouse with carrying a frameshift in this article, which will be a loss of function loss. We try to maintain this in the same genetic way as is found in patients. So it's heterozygous. There's always a correct wild type, correct normal. And we find that all these lines are viable, which means they live, which is true for your children also. Also, which is important for us experimentally, they can produce offspring. They can mate and have children, and both males and females. And so far, and this is at the beginning only, it's only starting. So we did not really analyze these mice in detail. We just try to have them and expand the colony and have more mice to look at. But we want to do a biochemical and behavioral analysis. So we want to find out if also in the brains of these mice, AGO2 binds to different microRNAs than it does in the wild type mouse. And this should be possible with these mice. So animal models like these mice, they are perfect models because they have the same situation. They have the mutation starting at the beginning of their life. They carry the variant. And in theory, this is all quite perfect. We can isolate the protein, we can look for associated RNAs. We can look for changes in protein composition because then once we really get the 3 wheels and the 5 engines or whatever, we can find this out. There's methods for that. And we can look for changes in brain structure and morphology, and we can look at synapses, do microscopy in any way. But mice are mice and not humans. So one phenotype of mice is they don't talk so much. I'm saying not so much because there is some communication with mice, but then that's a different thing. But they don't have this speech development like we do. They walk differently from humans, they've got 4 legs, so it's easier, and so on and so on and so on. So mice is a different thing in terms of the final analysis we want to do, but there are many, because mice have been in the biochemical research field for a long time, there are many standardized tests for mice where you can find out if their motor behavior is compromised, if their social behavior is compromised. Mice are quite social animals, and you can really measure this. So we hope that we can finally get more information. So this is some work that we will do, and this is planned for the next 1, 2, 3 years. See how fast we can go on with these things. So we want to find out which of the many molecular and cellular functions of AGO1 and AGO2 are altered by the mutations, in this case, especially for AGO2, which RNAs and proteins are altered, and how this affects neuronal function, and how this affects brain function. This is a whole series of different levels of research on the microRNA, mRNA level, on the protein level, on the systems level, where the whole neuron is looked at, and so on. And we hope to find something like outstandingly clear, where we think like, oh yeah, this is the thing that's going wrong in the mice and the neurons and whatever. And maybe one can use this to screen for compounds, for situations where this is altered, this is improved upon. This is maybe the idea. And this time I will not forget to thank co-workers. This is my group here. Deborah is also here. And this is the French group, Amélie and what is this? This is co-workers. Yeah.